Hi, hello everyone. My name is Al Hamadi with my colleague
Fahad Al-Tamimi.
from Qatar Science and Technology Secondary School for Boys, and our project is
Assessment and development of biotechnological testing methods for detecting bacterial contaminants in unlicensed artesian wells around Doha.
In the initial step of water analysis, begin by labeling an empty petri dish with your lab group and the water source. Then, carefully pour 1 milli of collected water into the dish. Label another dish as the control and add 1 ml of sterile water. Next, prepare the media by adding 5 ml of hot molten media to both the sample and the control dishes. Gently swirl it to mix without spillage. Then add cover to them to allow the solidification and incubate dishes at either room temperature or 37 degrees for 1 to 3 days. Finally, observe the plates and provide answers to the Petronet study questions based on your observations. This structure approach ensures systematic analysis without a research time frame.
Start by labeling media plates with your name or group along with control and specific areas such as the floor or doorway for sample placement. Leave the plates uncovered overnight in these areas to allow air exposure. Then replace the lids the following morning. Incubate the plates upside down at room temperature for 1 to 3 days or overnight at 37 degrees. And then observe the plates and see if there's bacteria or not. Module 2, we're focusing on detection and confirmation. First, we begin with observing color changes in samples for coliform presence and then using UV lights to confirm E. coli presence, and then adding indole detection reagent to color tubes and then looking for red ring development to confirm E. coli. This module requires specialized equipment such as UV transilluminator, UV safety goggles, and indole detection reagent.
Now we move on into PCR analysis. First, we begin by DNA isolation by sampling water containing various bacteria like E. coli, Serratia marcinus, and Bacillus subtilis, and also an unknown strain. We add 100 microliters of each water sample to the labeled tubes containing lysis buffer and incubate at 55 degrees Celsius for 5 minutes, followed by vertexing and incubating at 99 degrees Celsius for 5 minutes. We then move into setting up the PCR. We first label tubes with sample names and also add primer mix, extract DNA and PCR beads. And then we amplify DNA using specified PCR cycling conditions. And then we prepare an agarose gel and then load the PCR products into wells and conduct electrophoresis under specified conditions. We visualize the DNA bands using a UV transilluminator photograph and then we record our results.